Hi student, in this video lecture we will talk about one of the important topic from the genetic, that is about the Rh blood group system. As we have a lot of the blood group system, among the most clinically important blood group after the ABO blood group system, that is Rh blood group system. Now, this Rh blood group system, it is important because these will cause some clinical, some important reaction or some problem creates in the transfusion. So that's why these two blood groups that are very important. The first one, that is ABO blood group system, and the second one, that is Rh blood group system. Now let's talk about the Rh blood group system. So the first point, that is about the discovery. Now this Rh blood group system, it was discovered by Carl Landsteiner and Alexander Wiener in 1940. Now, this is about the discoverer who discovered the Rh blood group system. Now, why it is called as Rh blood group system? Now, the reason behind this is, so first of all, this blood group system, it was discovered in a monkey that is called as rhesus monkey. So the name of that monkey, that is Macaca mulato. So this is the name of the scientific name of this monkey. Now, and this monkey, the Rh blood group system, it is discovered. So that's why we call it is Rh. Rh mean rhesus monkey. Now, this is about the who discovered the Rh blood group and its why it is called as Rh blood group. Now let us talk about in which organism this Rh blood group system it is present. Now mainly this type of the Rh blood group system, it is mainly present in monkeys and men. So this is about the occurrence of the Rh blood group system. Now let us talk about the genetic basis of the Rh blood group system, that how the Rh blood group system it is formed, or what gene are responsible for its formation, Rh positive and Rh negative. Now look at see here the Rh antigen, these are controlled by a specific gene that is present. Now you can see here this is the structure of the chromosome number 1. Right in the chromosome number 1, this Rh blood group system, it is mainly controlled by 3 genes. Now the name of these gene, that is D, E and C. So these 3 genes are responsible for the Rh blood group system. Now these 3 genes, it is present on the chromosome number 1st. So right at the chromosome number 1st, these 3 genes occupy the 2 loci. Now you can see here the 2 gene E and C, it will occupy the 1 locus, while the D, it singly occupy the another locus. Now you can see here these 2 gene E and C, it will cancel the effect of each other. Only we have the D locus. Now this gene, it is responsible for the formation of the antigen. Now this D gene, it will produce the D antigen. Now D antigen, it is responsible for the Rh positive and Rh negative. Now you can simply here say that this gene, it is responsible for the production of antigen. Now if we have an individual that have a genotype that is homozygous dominant, that is capital D and capital D. So in this individual, the antigen or D antigen or Rh antigen, it is formed. Now we have a second individual that is heterozygous dominant. And this individual also the D antigen, it is produced, or Rh antigen, it is produced in this organism. Mean this organism is Rh positive. Now we have a third organism that is homozygous recessive. Now when such type of the genotype it is present in individual, no antigen is formed in this individual, and the individual it is called as Rh negative, because no production of the antigen occur in this individual. As the D gene, it is responsible for the production of the antigen. Now this antigen, in case of the Rh blood group system, we call it is Rh factor. Now Rh factor, it is actually as an antigen. Now this type of antigen, as in case of the ABO blood group system. Now this antigen, in case of the ABO blood group system, this antigen, it is present on the surface of the RBC. So the same cases, it is present here in the Rh blood group system. So the Rh antigen or Rh factor, it is also present on the surface of the red blood cells. Now if a person that have Rh antigen that is present mainly on the surface of the RBC. So if the person with Rh antigen that is present on the surface of the RBC, this he is called as Rh positive. While a person that do not have RR without Rh antigen, he is called as Rh negative. Now look it see here the case. Suppose we have a person that have a blood group A. Now why we call it is a blood group A? Suppose I have an blood group A. So why I call it I have an blood group A? Because on the surface of the RBC, the antigen it is decide the name of the blood group, that what type of the blood group a person have. If I have an antigen B on the surface of the RBC, so I have an blood group B. So I suppose an example that a person that have a blood group A. Now he have an antigen A that is present on the surface of his RBC. Suppose on this person have Rh antigen also present and at the surface of the RBC. Now this person know it is called it is an A positive. So the blood group of this person it will become A positive. So suppose if this person have antigen A and this individual does not have any antigen, Rh antigen does not present on its RBC, so its blood group it will be A negative. So simply the positive and negative that we will call it on the presence of the Rh antigen. Now so this point that is very contrast to the ABO blood group system. So you look at see here the natural antibody in case of the Rh factor is present. This means that the negative person who do not have Rh antigen also do not have antibodies in the plasma. So what does it mean? As in case of the ABO blood group system, if a person have a blood group A, so it must have antibodies B. But in this case, if a person have Rh negative, so it means that it do not have any type of Rh antigen. So that's why it have Rh negative blood group or Rh negative person. Now, if they do not have any type of antigen, it means that it's in this case they do not have any antigen as well as it does not have any antibodies. So in case of the ABO blood group system, if a person have antigen, it must have a alternative form of antibodies. But in case of the Rh blood group system, if an individual have Rh negative or negative blood group system, it means it do not have any antigen as well as no antibody is present and its body or its RBCs. Now how the formation of these antibodies occur? So this antibody it will produce by the exposure to Rh positive of a person that have Rh negative, but we will transfuse blood of the Rh positive person. So in this case the antibody it is from and pumped in the body of Rh negative persons, and it will take about 2 to 3 months. So such type of the antibodies that is pumped in the body of Rh negative on exposure to Rh positive person, such type of the antibodies this is called as acquired antibodies. Now we will see the percentage of the Rh positive and Rh negative person in different countries. So the first one that is in Africans. So in Africans all the individual almost that are Rh positive. Now while in Kenty and European whites, the percentage of Rh positive and Rh negative RS. So 85% of the individual that have Rh positive, while 15% of the individual that is Rh negative. So if we talk about the Indians, so in Indians 96.5% of the individuals that are Rh positive, while 3.5% of the individual that have Rh negative, mean they have a negative type of the blood group. So you can see here the universal donor blood group and universal recipient blood groups. So the first one that is the universal donor blood group, that is O negative. So why it is universal donor? Because it does not have any type of the antigen it of ABO system as well as the Rh blood group system. So on both type of the blood group system antigen it is present in this blood group system, so that's why it is called as universal donor. So any type of the antigen it is not present, so it does not interfere with the recipient blood group and with unable to stimulate the antibodies in the recipient blood group, so that's why it is called as universal donor blood group, that is O negative. While the universal recipient blood group, that is AB positive blood groups, so this blood group it does not have any type of the antibodies, as well as in the ABO A antibodies as well as B antibodies and RH antibodies, the all type of the antibodies it is absent in the AB positive blood groups. So that's why this type of the blood group it is called as universal recipient blood group. So these 2 points it is very important for MCQ's point of view. Now the important condition that is related to the RH blood group system, that is erythroblastosis foetalsis. Now this condition it is mainly developed in the fetus and are a newborn babies. Now this condition it is also known as hemolytic disorder of newborns or yellow baby. So you can see here in the newborn baby the breakdown of the RBC occurs, so that's why it is called as hemolytic disorder of newborns baby. Now you can see here the second name, that is it is also called as yellow baby, because in this condition the when the RBC it is breakdown it will form bilirubin, a product of the RBC. Now this bilirubin it will give yellow color to the baby, so you can see here the eye in the color of the eye whites it will give a yellow color, so that's why it is called as yellow baby. Now how this condition is developed? Now when marriage occur between the Rh positive parent mean father and Rh negative mothers, so when this marriage occur, so and the result a child is formed that is Rh positive. So at the first case mean in the first pregnancy when the birth of this child occur, so at this state the mixing of the blood occur. Now this blood it is called as fetal maternal bloods. So in this case the antigen that is present in the fetus bloods, so which will stimulate the antibodies in the mother body, and this stimulation of the antibodies it will take about 2 to 3 months. So in this month the antibodies that is mainly produced the fetus blood, so mainly the antibodies that is formed in the mother body. Now what happened? Now when the second pregnancy occur, right in the second pregnancy, so you can see here this is the uterus wall. So here the fetus it is develops. Now this a connection is present between the fetus and the mother body or the uterus. Now this connection through which the exchange of material occurs, now this connection it is called as placenta. So as the first pregnancy the antibodies that is formed in the mother body, now this antibody it can pass through the placenta and will enter in the body of the fetus. So the antibodies that is found in the mother body that is stimulated in the first pregnancy by the antibodies of the Rh mean Rh positive fetus. So you can see here this type of the antibodies that is IgG, so it is a type of the antibody and it have a special characteristics, it can pass through the placenta. Now when this antibodies it is enter into the Rh positive second fetus, so it will cause agglutination reaction, so which will cause clumping of the RBC of the fetus. So this type of the condition it is called as erythroblastosis foetalis. Now what are the symptoms of erythroblastosis foetalis? So normally in this case the clumping occur in the fetus body, so normally anemia like symptoms appear in the fetus. So what is anemia? Mean the deficiency of RBC occur in the fetus, so such type of the condition it is called as anemia. Now the second symptom that is jaundice. So actually when the breakdown of the RBC occur, the heme it is metabolite and form a product that is called as bilirubin, the end product of the RBC breakdown, so which will give yellow color to the fetus. So such type of the condition it is called as jaundice. The third symptom that is hydrofoetalis. Hydrofoetalis. So what does it mean? It means that the swelling of the fetus occur, edema like symptoms appear in the fetus. So such type of the condition, it is hydrofoetalsis, and it is mainly the symptoms of erythroblastosis foetalis. The fourth symptom, that is erythroblast, as the RBC level it is decreased in the fetus body. So to compensate this loss from the mother body cell, erythroblast, it is come to the fetus body. So that's why it is called as erythroblastosis foetalis. And erythroblast, it is actually an nucleated RBC that is produced in the fetus body. So this is mainly the symptoms of erythroblastosis foetalis. Now how this condition can be treated? So after the first delivery, we will inject these Rh antibodies as rogom injection. So it will contain the antibodies. So when this injection, it will inject into the mother body, it will call Cas agglutination type of the reaction, and it will help in the safe delivery of the second fetus or the second babies. So this is all about the Rh blood group system.